Phasmid vectors. Phasmids are also a combination of the plasmid DNA plus the lambda genome. And here the lambda genome includes the attachment site of the lambda genome DNA. So such a combination of plasmid DNA including the lambda attachment site, they are called as Phasmid vectors. Phasmid vectors, they can be synthesized by the insertion of the plasmid DNA into the lambda genome. And this insertion of the plasmid into the lambda genome, it is by the same mechanism by which the lambda genome insert itself into the host bacterial chromosome by a process which is called as site-specific recombination. If you remember, in case of lysogenic bacteriophages or cycle, lambda genome by site-specific recombination, it can insert its DNA into the host bacterial chromosome. तो इसके बाद जो lysis का step है वो temporarily inhibit हो जाता है और जो cycle है वो prophage stage में enter होता है, that is not immediately followed by the lysis of the cell. So similarly, the plasmids, they can also be inserted into the lambda genome by site-specific recombination, and this insertion, it is called as lifting of the plasmid. That is, the insertion of the plasmid into the lambda genome, it can be termed as lifting of the plasmid. And this step, it is reversible. Reversible means that कि plasmid जो है वो दोबारा से excise हो जाता है lambda genome से. So because of this reason, the insertion of the plasmid, it is considered as reversible. So such unique combinations of the plasmid with the lambda genome, they are called as the phasmids. Phasmids, they are very important in the cloning procedure because they have certain advantages over other lambda vectors, like here the DNA in the form of artificial recombinant DNA, they are more stable. When they are packaged inside the phage head, their shelf life increases, and at the same time their screening by the use of hybridization procedure, it is more easier as compared to bacterial or the other plasmids. So phasmids, they have certain advantages over other cloning vectors. And here we can quote an example of lambda ZAP vector as a phasmid examples. And this lambda ZAP, it can be used for the cloning of cDNA. And this lambda ZAP vector, it has certain advantages. It is a type of insertional vector where the foreign DNA, it can be inserted after treatment with appropriate restriction enzyme. So it is a type of insertional vector. After treatment with restriction enzyme, तो जो फॉरेन डीएनए का फ्रेगमेंट है इसको इसमें क्लोन किया जा सकता है और इसमें जो मैक्सिमम लेंथ है फॉरेन डीएनए इंसर्ट की ये 10 किलो बेस तक हो सकती है. So this is one of the advantage, and another advantage is that it may contain multiple cloning sites for different type of restriction enzyme. That is one cloning site for different type of restriction enzyme. So we can use different enzymes for cloning purpose. With reference to selectable markers, it contains gene for the synthesis of beta-galactosidase. We have already discussed the importance of beta-galactosidase inactivation. When foreign DNA, it is inserted within the gene that synthesizes beta-galactosidase, then it is inactivated, and it is called as insertional inactivation. And if there is no production of beta galactosidase, it means that the foreign DNA, it has been inserted. And at the same time, another advantage is that the foreign DNA, it can be excised from the lambda genome. In addition to that, these phasmids can be used to express the fusion or the hybrid proteins. And at the end, if the foreign DNA is inserted, then the RNA transcripts, they can also be synthesized. So these are the different advantages of the phasmid vectors.